quickly, today's plan will be a little bit of background information, what are process-related impurities, and specifically host cell proteins, what are they, why do we need to measure them, then place-based kit methods that we use and the limitations that come with those, and then my experience with Gyro lab technology, and then at the end, before the questions, I just wanted to share a little bit about my, troubleshooting experience and viewer profile data analysis examples. Let's start with the background information. What are the process-related impurities? These are type of impurities that are, derived from the manufacturing process, or cell culturing or downstream processing. For example, it could be, again, I will be mentioning about host cell proteins, but it could be host cell DNA, media components. This diagram that you see in the screen better explains the process-related impurities and their sources for the product development of, biopharmaceutical product development. So as you can see, the sources can be varied, and it can come in at any point of the development process. So why is this important? So when you look at from the risk assessment perspective, right, overall, all of these are possible sources, and they need to be considered and evaluated to understand the process-related impurity profile of the biopharma product. And specifically, host cell proteins, within these impurities, host cell proteins have an important place, and they are derived from the host organisms during biologics production. Host cell proteins are typically removed by the downstream purification process. However, if it is not removed enough and left around critical amounts during drug development, it could potentially impact the product quality, safety, or efficacy. So if that's the case, what are the accepted ranges, or in other terms, what is the threshold defined by FDA? Actually, there isn't a clear number, but as you can see, it basically says that remove HCPs as much as you can. So when that's the goal, most of the time, what we do in the lab is, for this reason, a lot of representative production process studies, and also we study a lot of worst case scenarios to actually document and evidence these HCPs were removed during the production stages, or development stages. So when it comes to measuring these HCPs, plate-based kit methods are, and the assay method, enzyme-linked immunosorbent assay, ELISAs, is the gold standard assay for HCP detection. And it provides a measure of the immunoreaction HCP in the product. And here you see a diagram of a, sandwich ELISA. This is the enzymatic reaction that takes place when we use, a plate-based kit. You can easily see the coating, capture, antibodies, and the conjugate interaction with the HCP or the analyte, and the substrate and the enzyme interaction that creates light. So using plate-based assays obviously has advantages, but it comes in with some limitations. And one of that limitation that I experienced in some of the programs that I work with or support is that dynamic range. Specifically, mostly Cygnus 3G, third generation kit is used a lot for these purposes, and their dynamic range for their standards are 1 to 100 nanogram per mil. And it uses, all pre-coated 96 well plates. So if you happen to run a lot of samples, or if you have a lot of replicates that doesn't fit in one plate, then you're dependent to use second, third plate or kit. And from there, I would like to talk about my experience with Gyro lab technology. So most of the data, as Rob said, that I will be presenting, and my experience is on the Gyros Explore technology, which is a smaller version of the Gyros Expand. Gyros to me provides a high-tech automated process for immunoassay testing. Testing is, as you can see on the first picture, testing is set up and run, very user-friendly manner, and the whole prep for me usually takes about, less than 30 minutes, about 30 minutes for a full CD run. And as you can see, you set up your plates for your samples and reagents, and then automation transfer that to a CD. And after that, once the run is over, by the end of the run, the laser-induced fluorescence detection reads out the results. And because most of the, once you put in all of your reagents and the samples in, rest of the stuff is all automated. So it saves a lot of time and also increases throughput because of the use of the CD.